Hello everyone, I'm Dr. Arpita. A very warm welcome to my YouTube channel. The topic for today's discussion is gluconeogenesis. Now, the word gluconeogenesis is a combination of three words. If we break it down, it is like gluco, the prefix gluco relates to glucose, it means glucose. Neo means new, and the suffix genesis means to generate, to synthesize. It means formation or origin. So in simpler words, gluconeogenesis means synthesis of new molecules of glucose from non-carbohydrate sources such as proteins or fats. So it is a metabolic process of producing glucose from non-carbohydrate sources like glycerol, lactate, and certain amino acids. It primarily occurs in the liver and kidneys to maintain blood glucose level, especially during fasting or low carbohydrate intake. Liver is a primary organ for gluconeogenesis, with kidneys playing a secondary role. Gluconeogenesis is regulated by hormones like glucagon and insulin. Glucagon is known to stimulate the pathway while insulin inhibits it. It essentially is the reverse of glycolysis, that is the breakdown of glucose, but it uses different enzymes to bypass the irreversible steps of glycolysis. If you can recall the process of glycolysis, there are certain irreversible steps. Those irreversible steps are not seen in the process of gluconeogenesis. Gluconeogenesis uses phosphoenol pyruvate, which is one of the intermediates of glycolysis, as the starting material. The major precursors for gluconeogenesis are lactate, amino acids from which form pyruvate or TCA cycle intermediates, and glycerol, which forms DHAP. The synthesis of one molecule of glucose from 2 moles of lactate requires energy equivalent to about 6 moles of ATP. So this is the account of energy that is associated with this process. Now we will talk about the steps of gluconeogenesis. So first and foremost step is what we see. Pyruvate carboxylase converts pyruvate to oxaloacetate in mitochondria. Oxaloacetate is then converted to malate or aspartate, which travels to the cytosol and is reconverted to oxaloacetate. So here you can see the oxaloacetate is first being converted to malate or aspartate, which travels to the cytosol and it's getting once again converted, that is reconverted to oxaloacetate. PEP or phosphoenol pyruvate carboxykinase converts oxaloacetate to phosphoenol pyruvate. Phosphoenol pyruvate forms fructose 1,6-bisphosphate by reversal of the steps of glycolysis. Now this is followed by another reaction where we can see that fructose 1,6-bisphosphatase converts fructose 1,6-bisphosphate to fructose 6-phosphate. That means you can understand that phosphatase enzyme is removing phosphate. Now this fructose 6-phosphate is getting converted to glucose 6-phosphate. Glucose 6-phosphatase converts glucose 6-phosphate to free glucose, which is released into the blood. Whatever we have just discussed, the steps are all here. You can see that the products, you can see the products here, the associated enzymes, and this is how the reaction occurs. how pyruvate gets converted to oxaloacetate. Oxaloacetate forms phosphoenol pyruvate, and then it goes on to produce finally glucose. This green side of it is showing you gluconeogenesis, while the pink side of it shows glycolysis, where you can see glucose gets converted to glucose 6 phosphate, fructose 6 phosphate, like this. So the pink arrows or the mauve arrows shows glycolysis, where pyruvate is the final product, and the green arrows and words show glucose as the final product, or gluconeogenesis. Now, if we briefly talk about its significance, we can see that it is crucial for maintaining blood glucose levels when dietary carbohydrates are limited, particularly during fasting or prolonged starvation. So, when a person is starving or is on a very low carb diet, what happens? The body needs glucose. Now, from where will it get? It will generate glucose from non-carbohydrate sources. Glucose is indispensable in our system. Our vital organs cannot function without it. So, if the external supply is low, the body has to compensate that. So, how will it compensate? It will compensate it through gluconeogenesis. It ensures a continuous supply of glucose to vital organs like brain, which relies solely on glucose for energy. As we all know, carbohydrate is the main food for the brain. Additionally, it helps remove metabolic byproducts from the blood, such as lactate from muscles and glycerol from adipose tissue. So, this is our brief discussion about gluconeogenesis. In this, you need to know what is it, where is it occurring, the steps of it, and its significance along with its energy account. So, this was our discussion. I hope it is clear to you. If you have any doubts, any confusion, please feel free to get back to me. Till then, thank you and happy learning.